group 1? I just want to see your distribution. Group 1? Okay. Okay. Now, group 2. What's under you are here? It's alright, it's alright, it's alright. Okay. We need to do this creatively, cooperatively, and attentively. Okay, otherwise it's going to get lost in translation somehow. If you want mask, you got some mask here. Okay, just a precaution because the cases in the hospital is on the rising now. We are quite crowded now. If you want mask, you got a mask here. Alright. Okay, listen up. Number 1, I need to create a new Google Space so that we can facilitate communication effectively between the two groups. Okay? Your lecturer from group 2 might not well for some time. So maybe I will take over for quite a week. We do not know yet. Just to be on the safe side, let's synchronize all of our communication. So what I need you to do is group 1. I sent the invitation to you to the new group, G1 and G2. You need to accept that so that you are in the group now. Number 2, group 2. If you do not put your name in the list, that's fine. Just come to the front, put your email here, your UTM email, and then put enter so that the next person can do it. So when you're done, I'll just hit the invite button. All of you will be in the group. Okay? Can you do that? Group 2? No? Put your email in here so that I can invite you to the Google Space.
Tulis email korang kat situ. Masuk email masuk email di chat. Limbat dalam ni. Kalau kita orang buat satu hari ni untuk tambah. Kita itu sewi. Supi. Pak grup tu pak. Mungkin tak pantas kan. Grup tu. Tinggi dulu.
Okay, just you can get the manual for
Dalam saya Tong stretching lah nak bagi. Kenapa?
So good. Are you done with your growth analysis lecture?
Group analysis lecture.
So group 2, are you done with your growth analysis lecture? Yes, no? Right? Very quickly. Define quickly. Very fast. Right. So if I ask you the curve in the form of sigmoid curve, alright. Basis of the curve. There's an out. That's only the first one. So that we call it. Stationary phase.
Duduk korang. Duduk korang. Duduk kan?
Duduk eh.
Duduk.
Not too early. So at least I just need you to be that.
Ni siapa je?
Group 2, I think we have learned it
Belakang.
better.
Tu baraya.
Okay. For group 1, I have taught first the introductory to crop physiology. What is it all about, and then the concept of growth itself. Okay. No mathematics just yet. Okay. But I think you have gone straight to the mathematics of it without proper introduction. Is that the message I'm getting? Right. Okay. So I think maybe we can do the revision together so that your information is up to date. Right? It's all the way from now. And for group 2, I think until in our Google Space, I will share the lesson that quickly so that it has been mentioned. So this is physiology lab. Well, this is not physiology lab, this is a teaching lab. The physiology lab is actually, you know where physiology lab is? No, it's on the corner, that corner. It's the largest lab in this domain. Right? The good thing is I am the coordinator for that lab. So please do as I invite. So the general rules apply to whichever labs that you get into, right? So make sure that your shoes are properly covered, okay? No slippers, no sandals with stockings, no, nothing like that. Check your friend. If your friend's not following the rules, yeah, you can be a snitch and come tell me. Right. Then if I hope nobody's wearing skirt or anything because that happened before. Skirt is fine, but down the knee. Alright? If you have long hairs, make sure it's tied up. Because we got a case before, the student got caught in fire. Right? You need to tie up your hair. You tie up your hair. Oh, you already tie your hair. tie up your hair. You can pause for beauty later. Afterwards. Nobody said that you were not beautiful. Right. Okay. So when you're with me, I'm kind of hardly not on time. Okay? So please be on time so that you can finish early. Do you have class after this?
Yeah.
What time? 11. See? You got time at 11. We cannot finish at 11. I want to finish early so that you can have extra half an hour to be prepared and go to your next class. So I want to be considerate for your timetable. So help each other to help yourself. Right? Okay. So, and then the goggles and gloves, these will be provided accordingly. And the special thing is just the mask today. I will bring this every week during the lab session. If you need to wear it, please wear it. Okay? Right. I'm not making any accusation to somebody else sick or anything. Just be cautious about things now. Right. For group 1, the lab report will account for total 20% of your coursework. Right? For group 2, if your lecturer wants to follow, there will be 20% as well. Right? So my style of conducting and doing lecture and practical, everybody should get A by default. If you don't get A, something not right with you. You see, the system is everybody at the beginning, your mark is 100. You do everything properly. You participate in the assignment, in the task given. Test 1, test 2, all the assignment default, 100%. Then you start to, because you have to work in a group, right? Then some of your group members got sloppy, some are not doing their work, you fight with each other, and when the deadline comes, no submission can be made, so deduction will definitely occur. So is that my fault or whose fault now? So own to it, okay? By default, everybody 100, okay? Right? So keep it 100 until the end. I can only not help you with the final exam, which account about 30 to 40% depending on the academic system at the end. Alright? So what if you manage to do everything, definitely you get A minus. Let's say that you screw up completely your final exam. Hopefully that's not the case. You definitely get A minus. You just decide, you know what, I don't want to do my final exam because I'm so confident with my coursework. A minus is enough. I don't need to brag so much about my achievement. That's fine. And guess what? I had a student like that. Yeah. So I think she only got like 5 marks for the final. 5 marks, no kidding. 5 marks for the final. But the carry marks for the coursework complete full. So she got a minus. So that happened before a few years back. Right. I want to watch it because now maybe a politician. Right. Okay. So and then about the lab report. Okay, for lab report, I'll just go quickly what is expected lab. In this later I will give you some sample from the previous previous year. I'll give you the sample how to do it for lab. So basically you are expected to have title, abstract, production, mathematics, result discussion, and literature cited. Have you been doing your lab reports this way?
Yes.
Yes? Did you follow everything? my advice to you is, because you are a science student, familiarize yourself with this scientific reporting system. Okay, title, abstract, production, material method, result discussion, and literature cited, or it's called INRAD. So, intro, materials and method, results and discussion. Result and discussion. So these are the fundamental of scientific report, but to make it better, usually we put the conclusion at the end. And then some more, title and abstract. Okay. You have all the AI tools in the world to help you to do this. So I don't want to see too much of grammatical errors. For example, abstract, that has to be in past tense. Okay? If you are not sure how to write it at first, I know I will be giving a sample from the previous year's student. You can use the AI to teach you, in your own time. For example, you can give the prompt to the AI. I would like to learn how to write an abstract. So ask the AI to guide you, because I can't teach you everything during our session, okay? Because that is rather a linguistic and technical things. I'm going to teach the science for you, right? And you can do the rest for the components of the scientific paper. So why this is important? Because during your final year project, you're going to do your thesis and project, right? It has to be in this form. And later on, when you go to pursue your study, perhaps, during your master or your PhD, you need to do publication. Everything is format along the way. So when you get familiarize yourself with this formatting from at young age, a lot better than wait until another 4 or 5 years later. Because from what I've seen, master student, PhD student who have not familiarized themselves with this INRAD system, they're going to have a very hard time in writing out, because they can't formulate. Even with the help of AI, it just locates scattered and sporadic the writing without having a good structure. So having an good command in English is of course kind of necessary these days. But having a good scientific structure, coherence, simultaneous, logical is equally important, because you want your experiment to be properly reported and reproducible. Maybe in the future people want to replicate your experiment because, oh, this looks interesting, the conclusion. Maybe something bigger can come out from it. But people need to validate your work, so they will have to refer to your materials and method. How did you do your experiment? This is why it has to be expressive and elaborate enough, so that even after maybe you have a long one after your demise, people can still reproduce your experiments. Meaning that you have managed to become a good scientist. Even without verbal instruction, just by looking at your report, another identical experiment can be done exactly like what it was done by you earlier. Right? Okay? So I don't have to set up projector now. So I think that's that with the reporting. Any question up to now? Discussion, reference, yeah. Question? Good. Alright, okay. I want to show you it, actually. Give me 2 minutes. I'll get this up right away. Because this is a newly formatted computer, so something might not be Okay, right. So I'll give you the sample now. So this is from okay, so this would be the first file for our chat space. So they can open. This should be the sample for the seed moisture content lab reporting. If you let me know if you are unable to see it, well, you should be able to get into the space first. There's only 32 members in this space at the moment. Shouldn't it be like, how many of you? 20, 27. Group 2, how many of you? 25. So there should be 50 something, right? What happened to the other 20? Who have not joined? For whatever reason? You need to join, okay? I'm serious about this. I don't want you to miss out on the lessons because you're not being attentive enough. When the instruction is given, you always join. Please join. Okay, two members, just join. Okay, anyway, if you are too shy to talk, just ask your friend how to help you, okay? Alright, group 1, did you manage to join, everybody?
Yes.
No?
Yes.
Okay, alright. I've put all your emails earlier today. Yeah. Right. So if you look at the samples that I've given to you, you can see that this particular group actually, well, they wanted 20 marks. I could give them, they got it eventually because this is not a fraction. You see, they have everything except one thing. What is it? Abstract. They not only they forgot, they actually refused to do it because they thought abstract is just, you know, what was that small paragraph doing at the beginning of my report. No, it's important. You need to do it. If you don't familiarize yourself to do it because you don't want to do it now because it's just too difficult or you think it's just not important, trust me, 3 to 5 years later it's going to be even more burdening, overwhelming, and more difficult. So get it over with. Widen your comfort zone. What is the problem for you now will become your routine in the next 5 years. Right? So they've got everything except the abstract. So let's see the introduction. So you can see that. I hope you are moving on your screen, okay? Because we don't have the projection now. You can see they have done the introduction for the seed moisture content. They have included some equation as well because that's what the exercise was all about. And then they also included the objectives. Actually, the objectives can be added at the end of the introduction. You know, objective, hypothesis, but you want to do it this way, that's fine as well. Right, and then they move to the methodology. Okay. I have actually deducted some marks here because it should be written materials and methods at first. Then they argue with me, of course, I didn't deduct the marks. So methodology or materials and methods are both accepted. Alright? So they kind of do all this reporting. The idea is you need to be visual, not to catch my interest. No. You make it visual so that it sticks in your memory. You know, humans are very visual. Right, we got 8 senses, right? How many senses you got? 11? 6? So, guess what? Your eyes actually not an organ. Did you know that? Actually, anatomically, eyes actually is brain. It's it's never a separate structure. Your eyes is actually a projection or extension of the brain nerve cord coming out, projecting. Yeah. So that's why visualization is a very good way to learn anything. You'll see easier than you would remember from hearing. You just saw that for a glimpse and then you remember it for a fact. But your friends have been chattering around your head for all the semester, you can't even remember one point. You see? Because, well, your eyes is actually a brain. It's just a brain. It's connected right to it. Okay? So go visual so that you can remember better and do all the leveling properly. Okay, like this group, they also took the instruments, right? Because you are in the process of learning. You want to be a good scientist, start with the good fundamentals. Every small thing, never a trivial. Okay, jot down everything because you are in the group. Okay, this is why you are made to work in a team. Maybe take duty, you know, rolling the duty. Maybe this week, the two of you will be responsible to take pictures and images, and the other three of you maybe deal with the experiment, and then you rotate next week, so that everybody will have different responsibility and know the bigger picture of it, right? I just want to add something, not at the risk of sound sexist or anything. Any groups with all girls? No? No, so you got the mix. The mix well. Okay. The mix well is fine. From my experience, when the group is all girls, that's the mismark that I got. Yeah, it's just a bit too drama in there. No, I only happened for one semester. It happened for quite a few semesters. Okay. No, I'm not saying that girls are bad. No, I'm just saying that you are a little enthusiastic in your own way. Okay. But you are still learning, and you need to inculcate the sense of teamwork. So don't go to Watson by yourself. Bring along your friends. Okay. And what happen if all boys in the group? Well, the first 3 labs never got submitted because everybody slept in, everybody got lazy. But when I have put up the warning and the sign, everybody about to sell it, magically 3 days later everything finished. See? Boys got that superpower. Yeah. But that warning didn't work for the girls. They want to stick with their belief and faith, whatever. So my point is, humans are dynamic, and so will plants. You will see later. Okay, so and there are results. You see there are reporting system here. You can see the raw data, and you can see they also use the Excel to visualize the data. You need to do this because later on when you deal with statistics, you have to do it anyway. Alright? So they have multiple data there, and then they started to discuss the finding. Okay, and they have some reference as well. So for this, how much they got? At first they got 18 because of the few mistakes, not mistake, omission, right? But this I have advised the group. Well, you don't have to send everything to me at the end of the semester. If you finish, you can send to me earlier. If something not right, you can go back and make corrections. So this is what this group did. It's kind of smart that way. They didn't submit to me at all at the ending of semester. They submitted a few weeks before the ending of semester, so they got 18. Some correction, then sent back to me, they got full mark. Yes, correct. I believe in second chance. Okay, sometimes you just a small omission, that's fine, right? So again, 20 marks, full marks is in your hand. You make the effort, you get it. I'm not going to penalize you because you are doing misspelling or anything. I just want to teach you. You need to be attentive, right? Because you're a scientist, right? Scientists need to be observant, right? If you want to be good science student, train yourself from the beginning, right? Okay, so that's that, right? And then literature cycle. Okay, it's all revision, not to worry. Right. Okay. I think that's all for the introduction to the lab safety and reporting system. Any question? Yes.
Okay.
The group 2, please don't feel shy to ask. Even though I'm not your lecturer, I'm still the coordinator for the course. So guess where your final exam question came from. Right? Right? Okay? Right. You need to try to ask verbally, that's why we have the space. You can ask them. Right. Okay, now let's do the growth analysis. I want to share something with you. Another thing. Let's see. It's the practical planning. You know what? I'll just snap this picture. Okay, I have a small disclaimer to make, okay, so that you are not going to blame anybody while we are going through this semester. You see, we actually have the full compendium of lab practicals for the whole semester. Usually at the beginning of the semester practical, we'll give the entire book to you, and then that's what we will follow. However, the book was not developed by me, and over the years I have noticed there are mistakes, there are irrelevant points in there, because it was created over 10 years ago. So now, this semester, I am in the effort to revise the old physiology lab practicals and out from the previous decade. So that's why you are not having that book compendium. So what will happen is, while we are doing our experiment, I will revise it, and that's why you are having these small handouts from week to week. So hopefully, by the end of this semester, I will manage to revise all experiments for this course, and then we will put it into a book, and everybody can have one working physiology handouts practical. Alright? Okay? So, right, let's look at the growth analysis. You see, did you get the image?
Yes.
Alright, so that's more or less the planning for this semester. So we are week number 1 now. Week number 1, we've done the briefing, the revision, that's good. Growth analysis, we'll do it in a week. And week number 2, which is next week. So next week is, you got your Hari Raya, right? So there's no class on Monday. So we'll go straight to, yes, go straight to the lab. Not to worry, consideration of leaf area. So leaf area here, I'll give you one example. What am I revising? In the old book, you will only rely on the graph paper and using the machine, like the conveyor belt, to determine your lift area, whatever lift that you take, then you run it in the machine will tell you at the end what's the area of it. Since you are now in the age of AI, I would like to include the AI way to do it. So I'm revising it so that in addition to traditional way, you will know the quick and mobile portable way to determine lift area as well. Right? Meaning that you don't have to rely on the computer or the big bulky machine to go along into the forest just to determine one parameter, namely lift area, which is not practical, right? So you are Gen 1.
Gen.
What? What gen are you? Z. Alpha. You're the Alpha.
Z. Millennial.
Group 1 told me they are, you are 20 years old, right?
Yes.
Okay, like you're older. Yeah, so it's okay. Even though we are different generation, I still can make you experience what it should be experienced by your generation. 20 years apart is just a number. I'm good at that. Even though I'm old, I can give you the good education. Yeah, 20 years. I was in UPM in 2003. No more. Where were you? Oh, yeah, that's correct. I started doing my part-time job in UPN in 2004. So I'm kind of archetype myself. One step away from becoming a fossil. Alright, anyway, that time flies. One day you will feel that. No, it's good. You learn agriculture, you know. You learn agriculture, you understand about plants. Then you will know what to eat. Because you eat agriculture produce, right? You eat the vegetable, you eat the beef, wagyu, whatever. All of those things, soybean, everything, is agriculture produce. So if you know how to produce healthy produce, you will consume things healthily. You will have a good and strong body for a very long time, right? And hopefully sound mind as well. There are many SEAL people these days. Okay, so let's move on to wait, let me finish this. So seed viability, and then we will have water and plant. You can see that there is some remarks there. So today actually we're going to start growing for your growth analysis hydroponically. So this is something different as well, because previously all students grow using sand. Oh, that's very messy and very, very dramatic. They have been doing it for 20 years. So now I'm taking over, so I can do experiments, right? Right. So we're going to do that today, and then hopefully after 2 weeks and after 4 weeks, you'll get double data, so that you can do your growth analysis, right? Right, and then we will have the first test on the week 6, and then the FCL and submission. Yeah, you see, week 7. This is what I meant. You don't have to wait until the end of the semester. I give you a chance if you want to submit early so that you know whether you get 20 marks or otherwise. Right? Right, and then we'll have the semester break, and then we'll continue with oxine and reorganization experiment, gibberellic acid and growth. This is called seed rejuvenation. And then we're going to have Christmas Eve special. That will be XCL. Yeah. And then we're going to have the second test of A. You should get to talk to your senior about taking my class. If you manage to talk to your senior, they might tell you that my second test will cause everybody to be asthmatic. You see, besides doing science correctly and soundly, I also believe people need to move, because that contributes to healthy life. So usually test 2 will involve you running. I remember that there was one guy. Okay, I know it was the legit name. During the test, he was on a wheelchair. So that he couldn't run, just collecting the answer for the friend. So my advice is, since you know you have already been given this sensitive for what's going to happen. If you have trouble running, you might want to build the stamina from that, because that will determine how much your group score for your test too. Okay, running is only 40% of it. You need to think as well. So have a good combination of people in your group. You know, some big guys, some small girls. That's a good thing. We'll make it fun. We'll make it fun. We'll make it fun. Okay. Oh, just one small question. Since we are in here already, you can see that the plan is to do it on the New Year's Eve, right? Yeah. This actually won't be in UPM. It will be somewhere else. So I'm telling you, I know you have class of the days. Is it a lot? Yes. What about group 2? Okay. If you can somehow coordinate with your lecturer or whatever after this to clear your day for that day, that would be better. Because the next day is New Year's holiday. I give you time to recover. Okay. Okay. So just for one day, just for one day. All right. Okay. Or maybe your lecturer wants to swap place with me. Also, okay. I'm going to ask this later. Okay. Maybe there are days because I think. What class are you having after this? What? Oh, okay. Alright, okay. We'll see how it works. We'll see how it works. Not to worry. We still have time for this because there are weeks I'm not going to be around. So maybe your lecturer can have my sessions instead. We'll see how it's going to go. Yeah. Right. Okay. And finally, that will be the instrument demonstration. So this we will do into it spectrometer, and spec is the instrument to measure chlorophyll. And then for the week 30, we do the instrument, another one for photosynthesis at ISO 800. We are very lucky to have the instrument because it's quite expensive. That one guy is costing $500,000. And we got two. Group 1 already know. Group 2, in case you think I'm not cut up for it, photosynthesis is not built, so the whole university using me to as well too when it comes to photosynthesis. Okay, and we have that machine in here. But we got two of the machines, right? And finally, week 14, SEL and full submission and whatever you factors you want to do on that day. Right. I see a lot of drama on the final day. Right. Especially for the Watson group. What happened? There was a blaming game. She's not talking to me. I'm not talking to her boyfriend. Too many drama. Please prevent drama. Okay? Right? No, you can have your drama whenever you go back to your college. Yeah. However, drama with me will have impact on your marks. keep it together. Keep it together. You can have fun among your team members. It doesn't have to be a bad experience, okay? It's in your hands. You determine whether this is going to be a rom-com genre of kind of movie, or it's going to be a thriller. No. Right. So very nice. Right. Okay. Let's do the final phase for today, which is the growth analysis. Okay. I'm not going to do this so much the reading. I'm going to explain verbally here. Then you can, because some of the things I'm going to explain are not in here, because I just revised this. I need to do it with you, then I will come back to here to revise properly again. Like I said, right, I'm in the process to revise this. So the idea is, let's see, what's on here. No, plus. Give me 2 minutes. Containers from I don't want to fold everything because it might get super wet.
Yep.
Right. Okay. Have you grown hydroponically before? I just need to see the ubiquitous of hydroponic knowledge. Who have not done anything at all with hydroponic before? 5, 6, 7, 10. Okay. Alright. Not to worry, that's fine. Come. Just need to learn. Okay? Right. Hydroponics means we grow the plant using a water culture. That is to say, it is under the science of growing plant soilless culture. Okay? By the traditional sense, you will grow the plant, the crop, using the soil. That has happened since the beginning of the human arrival on this planet, right? But with the advancement of science, people know that plants actually do not require soil to grow. Plants actually require nutrients for the soil to grow. So then, with the progression of times and science, the scientists learn what are the nutrients from the soils that are required by the plants to grow happily and healthily. Right? Do you know the periodic table of elements? Right? How many elements are there? 200. 200, that means you are in Neptune. We are on planet Earth. 100 what? 118. Okay. 118, and bear in mind, not all elements on the periodic table are found naturally. Okay, some of them are lab-made, right? from this 180 or 120, whatever number it is, how many are required for the plant growth? You know, from a seed, it can produce seed again. How many? If you look at your course outline, you're going to learn this anyway in the plant nutrition chapter. But we have a bit of head start so that you know you've got some perspective and good idea what's going on. How many? Who says 10? Nobody. So it depends actually on the school of thoughts. You see, science got a school of thoughts as well. Some says 16, some says 17, some says 18. Right? Since we are in UPM, we like to step on the facts, so we go with the 17. Right? What are those? This is the framework, okay? Carbon, hydrogen, oxygen. Oxygen. What else? So there's magnesium, calcium, sulfur, potassium, nitrogen, manganese, iron, zinc. Alright. 4, 5, 6, 7, 8, 9, 10, 11. What else? What did you Copper. Copper, 15. What else? What else? Chlorine. What else? Potassium. B is phosphorus. What else? What? This group. What is missing? So this is special. This is not all agree, but it's good to have. Nickel. Okay. Meaning to say, even without the use of soil, the moment these elements are present within the reach of plant, it's going to grow happily and healthily. And this is for the common plants that we know. You know, the vegetables, you know, all the fruit vegetables, leafy vegetables, they're going to grow happily. So that's what we're going to do with your growth analysis. We're going to use this DIY hydroponic chamber. Thanks to the lab assistant. They made this. We didn't buy this for you. They didn't go anywhere. I forced them to make this. Yeah. Alright. Even though they were all Watsons, but they behave. Alright. Okay. So this container is about 6 liters. And then we have some holes, and we have this a stretch of black covering for two reasons. Number one is to hold this little pot here. Number two is to prevent the algal bloom. You see, when you have the nutrient solution in here, if the water is exposed, the algae is going to bloom. You're going to get your unintended eutrophication, which you do not want that to happen. Do you know eutrophication? Yes. Alright, that's good. Right, okay. So what you need to do is we need to grow this using whoever know this, and you should know if you have read that thing. What's it? What's it? What's the Latin name for it? Pomia? Meaning that it's the cousin of who? It's the close cousin of who? You know this flower. Poaceae is grass. hour. It's a critical hour. Some people call it 10 morning. Morning what? Morning glory. Yes, yes. So Kangkong or water convolvulus, that's the vernacular name of it. They are of the same genus. In fact, they are of the same family, Convolvulaceae. Right? You've learned your botany, right?
Yes.
Right. What family is that? Family of what? How do you pronounce this? That means you are American. Okay, if you are British, because you are still have the sense of Commonwealth, we'll say this Poaceae. Poaceae. So don't feel weird if I pronounce it differently, because I didn't learn from the American people. At first. After that I did learn from American people. Poaceae, Convolvulaceae, Ericaceae, Araceae, Amarantaceae. So it ends with AC rather than RC. Right? Is it wrong? Well, both is accepted, but which is more correct? You know who invented? He says Poaceae. What language is this? That's not English. That's not English. That's not English. I need a better marker. The facility in this department is very sad. What do you call this? Yeah, I know. This kind of nomenclature, what kind of nomenclature you call it? It's got multiple ways to call this name. You can call it botanical name, scientific name, or Latin name. They are all the same. But when you call this what's this? Wah, you think lo langsung tak tahu ah? Buka. So this is called common name or vernacular name. So this means the same for this guy here. This tree means the same for this guy here. Get it correct. Okay? Even if you go to Alaska, you go to Nippon, you go to Moscow, still apply this rule. It's internationally accepted. Alright? But this still not internationally accepted. Okay, I don't know what else it's called. Right? Okay, so we're going to use the formula. You see the seed here. Diary. It's not small. It looks like you know terrarium. Got the pebbles in there, it looks like that pebble. Yeah. So what you need to do is, number one, you've got a container here. I need you to put the nutrient solution first. See, the nutrient solution has been prepared for you. This. Can you label the BC here? So this is called salt solution. There's somebody in there. boleh tak ambil the pengacau dodol malam? You can see that something is still unresolved. Alright, this is concentrated. Okay, so for the beginning, what I need you to do is you need to take some amount of the concentrated nutrient solution and dilute it. I need to get a better facility next week. Right. So, number one, get DIY box. Number two, we need fill 500 ml of stock solution. Which one is stock solution? This is stock solution. This is stock solution. You only take 500 ml first. Okay. Use this graduated measuring cylinder to get 500 ml, and then put it. Put the nutrient solution in here, and then carefully top it off with some water, tap water, but not all the way to the brim, just until it reaches 1 centimeter inside here. So that the idea is we get to moisten this. You don't want to fill all the way until here. No, not just yet. Okay. If your container is a bit loosened, you see the plastic is a bit loosened, you can get the tape to tighten it. Okay, so that all of your pots are in place. Okay, so after you have put your, so that's this is a cross section from the side. Okay, so this is your container, and this is your little pot. Right, so that's your little pot there. So you have put 500 ml of stock, and then you top it off with H2O until it reaches about this. That's all you need to do for this water. Right? How do you know it has reached? Your tissue now, so it's soaked now. Right? Once that has happened, you take this. What's this? Is it a dicot or one part seed? Right? We're going to learn later, but it's good to know it's a dicot. Take 2 seeds. 2 seeds, put in here. 1, 2. And then once you're done, we will bring this to the physiology lab, which is over there. Okay, I'll show you where. It's not that far. And we're going to put it on the growth rack. The growth rack will be properly fitted with LED, so that your kangkung or your ipomia can grow happily. So 2 weeks later, you can make your first harvest, hopefully, and then you can use that to dry the stem or the leaf or the root. Use whatever that you have to learn from the nutrient growth analysis for the data, the raw data. Meaning that you create your own raw data, right? Because you are in a group, you need to create a timetable among yourself. Who's going to check on the water? In the first week, it's no problem because the seeds are yet to germinate. But in the second week, when it has started to grow, the water level, the nutrient solution will start to deplete kind of aggressively. Right? So you need to have a duty roster among you. Who's going to check the water? Alright? I think maybe it has to be checked every 2 or 3 days. So if somebody already checked on Friday, somebody else check on Monday. Alright? Alright? So this is where your sense of teamwork will be tested. Okay? If you are super introvert, now is the time to break the ice. And if you find out that your friend is a bit of introvert and reluctant to participate, you should show your compassion and also your empathy to encourage your friends to participate. Because you also need to know how to deal with people. So all of this actually is the heads up for you, what the real life is going to be like. Some people are going to be difficult, some people are going to be very, very easy, some people are missing altogether. Let's see what's going to happen. Right? Okay, so why not? Let's do this first. Okay? Any question up to this point? Right. So you get your manual later, whatever you want to add or scratch later on your lab manual, you can do based on whatever that I have explained here. And also here. Alright? 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 Clear?
Yeah.
Clear? Clear? Yes. Just check water. Check water. If you're not confident to top off the water until it reaches the bottom of this pot, you can do it in the physiology lab. Because I know if the water is kind of full, your friend might trip on you and then the whole thing is scattered, right? Yeah, don't do that. If you don't want to do that, get the nutrient solution stopped first and then go to the physiology lab to top it off. Right? Okay? And then bring the seed with you. Oh, don't forget, you need to do the labeling. Labels your group name, who's the chief, and who's the what. Right? And the metric number is very important. Okay? And be careful, okay, this is glassware, this is not plastic. Okay? Please do not break anything. Right, I think that's pretty much it. Any question? Oh, and finally, after you're done, but that can be done later, we need to show the plastic. After you have put it on the growth rack, you need to put this one liner on top of it to trap the moisture. Yeah, just cover it to trap the moisture so that your kangkung or ipomia will go faster, germinate faster. You can use the tape later to tape it, this side and this side. Okay? Alright. Right. Okay. So make sure this is not, you are not growing rice. I said you are not growing rice. So this shouldn't be all the way soaked here. You need to be able to control the water level. Okay, because this is DIY. Right? Okay. If there's no question, I'll be here. You can ask the question. You can swarm about and do it. Alright, you can get yourself. You can do everything.
Ada gambar gila. Dia gila. Label ni. Mana promo?
lab ni ambil tempat kan. Listen. You take this part, you can use this speaker. You can use this speaker. That makes sense. Where is the... No, I'm looking for this thing. Yes, you need to stir it. Oh. Use that. Follow me. Wait. Hold it. The group will be missing train for a moment. Group number 2. How many groups are you in your classroom? 5. Okay. So our group also 5.